Good afternoon, everybody. I know it's a little small today, so we'll wait a minute or two before we get going. I know it's like just a couple weeks out from the holidays, so maybe everybody's just getting tired or looking forward to that and taking a week off. So we'll wait a minute or two before people start trickling in. Rukia, if you can hear me, can you maybe blast out another email if possible? Maybe people are in the old meeting, they may not have realized that we switched to the webinar format. So just in case, because I know normally we have Marguerite and maybe her students, so maybe they just clicked on the wrong link.
Okay, I'll do that now.
Perfect, thanks. But glad to have Pratik here, Dr. Hankins, Nick, Monique, Levi, Katie, Debashish. Hey, Captain Yamamoto, good to see you. Dr. Wu. We're trying this new format, and also Jim, how are you doing? I know we're trying this new format, so please be patient. I think we're moving forward in the future. This is just going to help facilitate questions and answers better, especially as we to hopefully everybody's agreement, trying to get more reach for HiPAM, and this allows us to reach more of the public. As much as we really enjoy our really informal sessions, I think this is a change that we need to make to make sure that we can get more reach. So with that, we'll wait one more minute, and then let me start to share my screen so we can see the updates for this week for HiPAM. Perfect. So we'll get going, and hopefully everybody will trickle in. So good afternoon, everybody. We are on the 12th of November, and here's our update. So national snapshot. Unfortunately, again, I don't think this is surprising to everybody on our meeting today, because I think we all pay attention. We are unfortunately continuing to trend upwards. And now, our last week, our 7-day average was still under 100,000. Now it is firmly above, and I believe Sunday, no wait, what day is it? Thursday. Earlier this week, I apologize, the days blur together. Earlier this week, we had another high of over 140,000 on our daily case for the nation. So unfortunately, that's going to contribute to a continuation of our increasing trend of our 7-day case average. Our numbers last week. Our 14-day, our 7-day average was 92. On that day, we had 156 cases. 7-day hospitalization was 6.3, and fatalities was 1.3. This week, Thursday, this is pulled from yesterday, so it's a day lag. Unfortunately, we are trending upwards again in terms of our 7-day average. Now we are back at triple digits. Hospitalizations have continued to trend up. Fatalities trend down, but once again, they're on a daily level and maybe even a weekly level. Our 7-day average fatalities will be the most variable because of what we understand COVID and how it affects our certain populations. So once again, increase from, you can see that it was trending down, bump a little bit, trend down. Now we're seeing increase and a slight trend down in terms of our 7-day average. We'll talk about this a little more when we get to our projection slide, but really last week was when we said we're at an inflection point with our increasing, and then this week and also next week, I think we'll be really careful to monitor the major impacts from the 15 October opening, tier 2, more utilization of the restaurant card, etc. And Halloween. So we're really at an inflection point now, but you can see 92 cases from last week for the 70 average up to 100. Obviously fatalities is very fluctuating. Marguerite, thank you. Rukia, if you could send the link to Yosh and Tiana so that they can join. Hospitalization, slight increase. Once again, still well within our capacity, but we understand there's a lag in case, with the hospitalizations relative to cases. So that's another data point we are going to look at. Good news is that ICU utilization for COVID is still, while it did increase by 5 from last week, it is still much lower than where we were a month ago and 2 months ago. So we will continue to watch that if our cases continue to increase locally. Our percent positivity increased slightly for a state from last week to this week. So once again, continue to watch that and see if there's any increases due to travel. And then our test turnaround time is slowly, it's still holding steady. If you look at the pure volume of numbers of tests that are returned within a 24-hour period. However, relative to all of the tests that were conducted last week, because this is a week lag, we're not as good as we used to, at least in terms of October 11th and October 4th, where we had high 80%, 90% in terms of return under 24 hours for our tests. Once again, this is important as we had opened up on 15 October. Our other experts from the state identified Texas, Arizona, and California as 3 major states that contribute, to our tourism, and a lot of our Ohana that live here visit these states. So we continue to watch these states in particular. They're all trending up very clearly. So once again, even with our 72-hour pre-test and with our surveillance, is still something to keep in mind, especially as we don't have a lot of information in terms of our surveillance testing for that post-arrival second test. And then with potential opening up to travel to Japan, I apologize for not knowing the exact date. It is once again clear that Japan as a whole is now trending up. They are now in the thousands for their 7-day case average. So once again, introducing yet another pot of travelers to Hawaii coming from a country that is trending up. Just something to note. One thing that we hypothesize is that Japanese tend to be, and a lot of our Asian counterparts tend to be very careful about their behaviors and compliant with mask wearing and very safe with their behaviors. So while this is concerning, there are other nuances that we are aware of. Let me see if Levi is on the chat. Rukia, if Levi's on,
yep.
If you can.
Can you hear me?
Yes, we can. Go ahead. Take it away, Levi.
Okay, great. Thanks, Thomas. So when we're looking at the forecast for this week, again, it's a little hard to tell. So basically what we're forecasting out right now is around a flat curve, because it really could be going either way. There is obviously this downtick in these last couple of days, but again, we're still on the precipice with all this travel going on. It's hard to tell if this is really a downward trend or not. So right now we're kind of projecting flat to slightly up. So when we're looking at that, that's still closing in on around the triple-digit case count. We don't really expect to be going a lot below that at the current levels right now. And still, from a hospital bed standpoint, we are lower than we have been in some time. We're still at relatively comfortable levels, but this could change quickly again, especially if our case counts start to really dramatically go up. So right now, just kind of forecasting a slight increase, but again, within the next couple of days, this obviously could change. So we're watching this really closely as we with all this travel coming in.
Perfect. Thanks so much, Levi. Monique, I believe that.
Hi.
Please take away. So I have this slide, and then when you're ready, I can hop to the next slide.
All right. So we just incorporated into the model the travelers. We divided them into resident and tourist. We have a new category in the model for the tourist, and so this is so the red the orange dots are the real count, and the red is a curve. That is assuming that 1.5% of the travelers go through the loop of the pre-test program and are actually exposed. So we divided them into different expose days because we broke that down into from 1 to 14. And this is the 1.5% was just the one that would give us the best fit to the data. That's a large percentage, but this is assuming that there is nothing else. We see a little bump that was due to the fact that we changed the transmission rate with the tier 2 reopening. And that's also a little bit the explanation why it's going up. So if you can go to the next one. This is the same, using the same model, but assuming that we did not open up for travelers. This is where what the model is projecting if we would not have started the pre-travel test and left it as where we are. Just if I can just add a little bit something, it's unclear whether it's the travel or not. That could be one explanation for the spike. I also realized this morning that there is another correlation that started 14 days ago, about a little more now. It is the restaurant card. That's when people started receiving it, and restaurant and gym have been labeled as one of the big vehicles. So that's something to think about. Thank you.
Perfect. Thank you, Monique. There'll be time for questions for Monique and Levi in a little bit as soon as we finish our highlights. So two highlights for this week, especially I think it's the first one, SARS-CoV-2 in the US military, lessons for civil society. I think that's important, especially given that this is around the time that we're going to start to see college students coming back home, because there's some strong tie-ins to potentially college dormitories. And then I thought this was super interesting, and I will defer definitely to our clinicians in this group to explain more and to answer questions, but pre-existing and de novo humoral immunity to COVID in humans. So this first highlight, New England Journal of Medicine, is an editorial. So some recaps before we potentially talk about impacts to Hawaii specifically. So it feels like forever ago, but when there was that outbreak on the USS Theodore Roosevelt, about 77% of all of the sailors who were tested at the initial diagnosis were asymptomatic. And of them, 55% did not show symptoms at any time. And the reason why this is important is, it's a demographic that we have identified in the past few months as being asymptomatic spreaders all around the world. Obviously, you see increased spread in close quarters, but we learned a lot from this particular natural case study. And then also there was another example in the military where at the US Marine Corps recruit depot, they tried to implement a phased quarantine. So the recruits quarantined for 2 weeks prior to reporting to basic training. And then they had a 2-week period where they were quarantining within the platoon. So of the 1,800 recruits, within 2 days, there were 16 positives, 15 asymptomatic. And this is with a modified quarantine where they did test upon arrival. And then at that second test, there were still more positives. And then on day 7 of that 14-day supervised quarantine, they had more Marines test positive. So some takeaways specifically would be, it is important to remember that, we have this 14-day quarantine, but, it can extend beyond that window in terms of people testing positive. We're still figuring out truly how long someone can transmit the disease. And we've seen 1 and 2, individual cases where it's much longer, but as an average, it's within 2 weeks. But this just reminds us that with COVID, there, exceptions can occur. And then with UH and our dormitory settings and potential for students being having cluster situations and super spreaders, we've seen that with this demographic, it is possible. And it's important to note that, on the Theodore Roosevelt, it was a very controlled environment, with our dormitories here, and this is just my opinion. I think that it's not as controlled. So the potential for spread in this demographic to the rest of the public is increased compared to a much more controlled environment in a military setting. The other interesting, and to me as a lay person when it comes to this type of the science field, I quoted, there was an accidental discovery by researchers at the Francis Crick Institute and University College London while testing the performance of sensitive COVID-19 antibody tests by comparing the blood of COVID-19 infected donors with that of those who did not have the disease. And basically what they found was that there were samples from some non-infected donors, specifically children, apologies for the typo, where they contain antibodies that could recognize both SARS-CoV-2 and other possible coronaviruses. Earlier in this response, we were trying to hypothesize why children were less impacted. Dr. Hankins, I remember we had this conversation, and this might be because children under the age of 6 or 7, typically, due to where they interact, they're exposed more to coronaviruses. It is not evidence in that we can say that this is why there's fewer transmission or infections, but we're at least starting. This is a hypothesis that I found interesting. I know that this is when I would open up to Q&A and we would just have a discussion with this new format. If you have questions, please type in the Q&A, or if you raise your hand, we can give you the ability to ask your question verbally over the mic like we normally do. So please, if you have questions on Monique's modeling, Levi, or the COVID highlights, or the data trends, definitely now is the time to ask before we invite Claire, Marguerite's grad student, to present for our deeper dive. Rukia, I'm not able to see people raising their hands, sorry, with my screen. So if there's anybody raising their hand, please go ahead and give them voice access, please.
I don't think anyone
There's no
is
questions?
raising
Okay.
their hand. Yeah, I don't think there's any questions.
Okay, well, let me look at the chat. Okay, sounds good. So I'm going to, Claire, I'm going to hand over control to you, but Marguerite, if you want to introduce your grad student, let me just make sure that I did give, Claire, if you can just make sure that you can control the slides. I believe I gave you control, so. And then I think you're muted.
Yeah, I think you're still muted, Claire. Okay, so hi everyone, thank you. I'm really excited that Claire is going to be giving our short deep dive today. She's a second-year master's student in microbiology and got really interested in diving in to help with the COVID-19 pandemic in Hawaii. So she's been looking into genotypes of SARS-CoV-2.
Yes, thank you for that introduction, Dr. Butler. I don't know how to go back in the slides. Let's see.
Let me, oh, hold on. Okay, let me give you,
control? Yeah.
sorry,
I took it back just so I can go back. Claire, you should have control now.
Yeah, I could progress, but I wasn't, I couldn't go backwards. Yeah, thank you, Dr. Lee. All right, so thank you all for having me today and listening to my talk. Today, I'll be talking about the evolution of SARS-CoV-2 over time and how the virus is diverging into different genotypes. There you go, okay. So SARS-CoV-2 has an inherently high mutation rate that provides the opportunity for the virus to alter its genetic code and evolve. So here's a zoomed in portion of the family tree or the phylogeny of the virus, where each branch of the tree represents divergence as a result of mutations. So as we move to the right, we are moving forward in time. As you can see indicated at the bottom, we have the months there. So there are some lineages that die out, while others persist and sweep through the population. So those would be the branches, the longer branches that are present in the later months. And so strains or genotypes are defined as variants with a set of mutations that sometimes will cause a functional change, but it's not always required that there is a functional change there. So scientists have been genotyping the virus, and the presence of these genotypes vary across different continents. When compared to the reference genome of the first sampled sequence in China, there were 6 mutations that were detected across all continents. Of the virus into these multiple populations through long distance dispersal. So this comparison did, however, detect greater differences across certain continents. As you can see here, we have 242 in Europe, 224 in Asia, and then 154 in North America. So this portion in green shows mutations that are detected in at least 2 continents, while on the outer pink regions, these are mutations that were unique to the continent. So we know that there's a variation in the number of mutations across different populations, but do the different genotypes matter? How do these mutations actually alter the virus? So here's a map of the genome of SARS-CoV-2, which is a 30,000 base pair long RNA virus with multiple gene regions that code for important functions such as viral replication and host entry. The genetic code determines what type of proteins are generated, and I would like to highlight the S gene here, shown in green. This codes for the spike protein that binds to the host cell receptors for entry. And I'll be talking more about this protein further on. In addition, it's important to note that SARS-CoV-2 has an inherently high mutation rate of 10 to the -3 to 10 to the -6 mutations per replication cycle, which allows the virus to evolve relatively quickly. So a little bit of background, the genetic code, whether it is DNA or RNA, codes for proteins in triplets. There are degeneracies typically within that third position, so some mutations do not change the coded protein or are silent. These changes are also known as synonymous mutations, as they code for the same protein. However, sometimes we have mutations in the genome that cause a change in the protein, and these are known as non-synonymous mutations. So SARS-CoV-2 has a spike protein which is involved in host cell entry. It's made up of 3 identical subunits that can either be in an open or a closed orientation. With more of these subunits open, there's a greater chance for the virus to infect a host cell. And studies suggest that at least 2 of these subunits need to be in the open position for the virus to bind to the host cell. With the mutation of the spike protein in position 614, resulting in a change from the D to G variant, these subunits have been detected in the 2 open state at the highest frequencies. Without this mutation, the spike protein is observed in the all closed or the 1 open state at higher frequencies. And this D614G mutation is a result of a single point mutation that is non-synonymous, and it changes the actual structure of the spike protein. So we go from an aspartate to a glycine. And there are thousands of mutations that have been detected in SARS-CoV-2. And here are the locations of some of them. And the actual protein structures of the virus are actually very beautiful to look at. And these frequencies of the mutations have been mapped across the genome of SARS-CoV-2 to see what genes they're associated with. So are these mutations truly random, or does the same mutation happen repeatedly? The D614G mutation is important to highlight due to the resulting increase in infectivity. This variant started at low frequencies at the beginning of the pandemic, and after introduction into Europe in mid-February, it swept through populations across the globe. So it seems like this mutation is helping the virus succeed as it increases the transmission to be 10 times more efficient than the other variant. When there is a mutation that's very beneficial to the virus, it is likely to spread and be prevalent in multiple strains of the virus. So that was a good example of positive selection, where we can see an increased frequency of mutation above background mutation rates. So when mutations first arise, they are rare, but advantageous mutations will start to dominate, which is also known as positive selection. So we can see here on the left, if we imagine that these are 4 different strains, we see that there is a higher frequency of the mutations, which are indicated by stars. So these will start to have a higher prevalence in the population. And the other important form of selection is purifying selection. There are mutations that occur that are detrimental as they change regions of the genome that are important for proliferation. These mutations will not be passed on to further lineages, as this strain indicated at the bottom here, or covered by an X, will die off, and you will not see that virus proliferate. So multiple groups of researchers have run analyses to detect selection across the different genes of SARS-CoV-2 based on the rates of non-synonymous and synonymous mutations. Selection can be used to find important gene regions where positive selection is expected to result in variants with greater fitness or that have some sort of potential advantage. Detection of purifying selection, on the other hand, will provide information for which regions are highly conserved due to importance of viral proliferation. And so far, researchers have observed positive selection in the spike protein, with one example being the D614 gene mutation, and purifying selection in gene regions that code for proteins required for viral replication and the structural membrane. And these selection analyses are very important as they provide clues for which functions or parts of the virus are important for viral success. It's an evolutionary analysis that tells us what gene regions are important for the virus. And if we know what mutations are of importance, it may become important to know what strains our patients have. And there are complex patterns of different genotypes that are detected around the world. There were probably multiple introductions of various strains early on that continue to evolve within each locality as we see multiple genotypes present with different geographic locations. Researchers are tracking the success and spread of these genotypes through time, and we can now begin to correlate the evolution of genotypes with rates of infection, at least at a population level. And this is actually what I plan to do with the local community. And here I have the phylogeny of the isolates we have here in Hawaii. And these sequences are from March, and they are unfortunately the only sequences that we have from our state. With sequences of the isolates that are currently in circulation, we can test for selection or random evolution to determine if genotype matters within our local population. Because we are a US state of relatively low case incidence, it would be interesting to see if the evolution of SARS-CoV-2 varies within our community compared to localities with high incidence. And replication cycles are observed with host transmission, and exponential growth of the virus presents ample opportunity for mutation. We can see that if increased transmission of the virus changes the evolution of the virus. So what exactly is happening there? And the lower case positivity rates may potentially allow for more competition between the strains that are present if there are fewer strains circulating within the population. So if the virus doesn't have as much of an opportunity to evolve and mutate, we can see is there a difference in the evolution of the virus within our population. And we'd like to thank Dr. Butler and the Butler Lab, as well as Dr. Vivek Nerurkar, who is head of the JABSOM Tropical Medicine CLIA Lab, for his collaboration and allowing us to have access to sequences from the state of Hawaii. as well as Dr. Masato Yoshizawa for collaborating and allowing us to use his lab space, and the HiPAM group for allowing me to talk today or share this information with you. And with that, I'll take any questions. Sorry, my light's on a timer.
Thank you, Clara. Let me check the Q&A. So no questions yet. I have a couple questions. Let me try to go back to this, and hopefully other people will have had the same question. So I'm surprised that Australia has over 1,500 samples compared to the United States. Is this just due to them being more on top of it when it comes to studying this particular part of COVID?
So the US has a lot more samples than the 910, but this group was just looking at a subset of the samples that we have.
Oh, okay.
Yeah.
I believe that Australia has a project to actually sequence every single case.
Wow.
So that's probably why they have so many samples.
That's awesome. Claire, I think you have two questions in the Q&A, one from Andrew and one from Dr. Shiba.
Okay, awesome. So do these genotypic differences result in any major difference in phenotype? When should we be concerned? Okay, yes. So some of the differences in genotype do result in a change in the phenotype. As I mentioned with the D614G mutation in the spike protein, that actually changes the structure of the protein. And as I mentioned, it causes two of those subunits of the spike protein to be in that open position, which increases infectivity of the virus. And they're looking into more regions or genes that have mutations that actually may change the phenotype, but this is the first kind of mutation that they noticed was actually causing a really big change in the phenotype of the virus. And then Monique, does the climate affect the mutations? So it is possible that there is a correlation with mutations or the genotypes that we observe in different climates with different populations at different climates. But so the figure that I showed earlier with the number or the frequency of mutations within the different continents, I believe it's the third slide. And so that group also did look at climate to see if there was a difference there. But there aren't too many studies that actually look into that. So I'm not really sure about how much of an effect it has for mutations.
Claire, one more question from me on the topic of climate. In Hawaii, we're very unique in that we always get visitors and travelers from all over the world because there's always someplace that's cold. And does the fact that we have travelers from from the northern hemisphere, and then maybe some mixing with theoretically some from Japan, would that potentially increase frequency of mutations when you have interactions from people from different continents in Hawaii?
Yeah, good question. So if we have the introduction of multiple strains, then we're essentially allowing for different strains of the virus to evolve independently. But it depends on how fit these strains of the virus are. So if one strain is more fit than the alternative, then it might be outcompeted by that initial strain. But there is definitely that opportunity for those two strains to evolve independently. So then, we do have to be aware of the consequences of that.
And basically, we would be the biggest petri dish ever if we have all the strains coming to our shores.
And that's including our returning residents, right? So, go to whatever state they go to and then bring it back. So obviously New York and some of these other states will experience something similar. But so
far it's been I don't think there's much evidence for recombination. So like with HIV, I think one of the scary things is if you have multiple infection in the same host, you could actually have potential for recombination. But so far, doesn't seem like it happens in SARS-CoV-2, and let's hope that it remains that way.
Yeah. Claire, you have a question from It's Fun.
Oh yes. So in addition to mutations that increase infectiousness, are there mutations that make the virus less dangerous? If yes, the intriguing possibility would be to combine them, since the less dangerous, more infectious virus would take over. Yeah, that's an interesting question. There are mutations that occur that do make the virus, I guess, not less dangerous, but they do change, the structure of the virus, so it may have an impact on infectivity. But yeah, that would be interesting to see, the combination of, the two, I guess, more infectious and less infectious mutations, and see kind of like how that works out.
And then Dr. Hankins.
Yes. Given that COVID-19 is zoonotic in origin, is there any evidence that continued zoonotic interaction could play a role in future viral evolution similar to flu? I think so, because anytime that the virus will jump from one host type to another, there has to be, or it has to be able to infect essentially that next host type, right? So typically the virus would need to evolve or have some sort of mutation that allows for that jump to happen. So, changing the, or mutations within the virus, as it would move from host to host, there is, that greater potential for the virus to evolve within that new host. So, we don't really know what the impacts of that would be, but just providing it a different type of environment, gives it the potential to mutate in a different way than it would within the human host.
Yeah, I guess I should say I'm not a virologist. I'm actually an evolutionary biologist, but I know that what they're looking at is the first is the interspecies jump, as Claire said, and then the other event that is important for something to become an epidemic is the mutation to allow the virus to jump from human to human host. Like if both of those barriers are crossed, then you have the potential, because presumably there's lots of zoonotic crossings like that happen all the time, but then if that second level doesn't jump. But I mean, we've had how many SARS viruses, so yeah, this is probably going to keep happening.
Yeah, it's definitely terrifying. I see another question from Francis. So at what point will mutations affect the effectiveness of vaccines? So that's a really good question. This is totally dependent on what the vaccine is actually targeting. So we have, depending on the different structures of the virus or different functions that the proteins are, used for or associated with, we can use these as targets for vaccines, because they essentially will prevent the virus from entering the host or prevent the virus from replicating. So if it is within a gene that is essential, or that we're actually targeting for the vaccine, then mutations there will be it will actually decrease the effectiveness of our vaccines. So that's why we need to be careful with what we're actually targeting. Yeah, to make sure that the vaccines that we produce are going to be long lasting.
So I have a question that kind of is similar to that, and I posted this in the Slack this morning or last night. For our smarter hardcore scientists in attendance, at least with the mRNA vaccines that are being developed right now, do you have any idea of the possibility of someone being inoculated with that type of vaccine and still potentially being able to get COVID, and then maybe like it's fun, as San Francis asked, so there's mutation, but also there's then still a possibility for a transmission of COVID with someone who did get, you know, the vaccine?
Yeah, I'm not too familiar with the mRNA vaccines, so unfortunately I don't think that I can answer that question too well. Yeah, Dr. Biller, do you have any comments on that?
Sorry, the question was whether mRNA, whether you could get reinfected. I don't know. I mean, I think it depends on what type of immune response is generated and whether it's a barrier to cells infecting the upper respiratory tract. I think the jury's out on that one.
So I know we have some clinicians out there who maybe this is more their research interest. So if you have a response, please raise your hand, and we'll love to hear your thoughts. So, but if I can summarize from what we're hearing so far, it's still too early to tell whether or not someone who gets vaccinated will. So if you get vaccinated, you know, you're more protected than not vaccinated. But we can't say for certain that if you get vaccinated, you're not able to transmit the disease. I think that's something that we're, at least based on what I'm reading, we're still unable to make that determination, and at least communicating with the public that the vaccine is not a panacea, and that it's not going to magically stop future transmission, at least based on what we understand how most viral vaccines work. So let me see if there's any.
Well, the problem is that, I mean, RNA vaccines have never been used before. So this is a first test case use.
Great point. Great point.
But I'm very, very happy that Dr. Fauci is happy about it, and that that the initial results have a 90% you know, effectiveness. So yeah, I think everyone's waiting to see what the data really look like and what more of the results look like.
Perfect. Thanks, Marguerite. Rukia, if you could, I think Dr. Hankins is raising his hand.
Yeah, I was just going to say, absolutely, a 90% efficacy rate means that 10% of the people who are vaccinated are still susceptible to infection. What we don't know is whether they'll still have transmissibility, whether the disease impact will be reduced somewhat in terms of severity, because we certainly see that, you know, every year with, for example, our flu vaccines. And yes, this is a different virus, but so folks with the flu vaccines can still get the flu, can still transmit the flu. We know that, but again, this is a new technology. I'm not sure that it's going to necessarily change some of those fundamentals, though.
Thanks so much, Dr. Hankins. And then Dr. Wu has a comment. Getting a vaccine does not equal preventing someone from being infected, but to prime the immune system to fight off infection more effectively. So once again, all depends on the immune response to the vaccine. I think Marguerite had a really good point in that this is a different type of vaccine that we're used to. And so we don't have as much data points and as much data to draw upon historically. So it's, it's a wait and see. It's great that with this new technology, we have a vaccine at a much faster timeline. And I agree that if Dr. Fauci is happy, that's a good sign. But, you know, once again, tempering expectations for our local public and helping them understand the limitations, the strengths, and also limitations that this type of vaccine will bring will be important. And hopefully, since Dr. Wu is on here, maybe not today, because I don't want to throw you under the bus, but maybe when you have time, if you wanted to pitch more about local vaccine efforts. I know you're sitting on that, and I'm not sure if Dr. Hankins, if you're sitting that as well. But it'll be great to have more insight on, you know, logistics as well, because we a previous talk maybe a few weeks ago focused on not just the vaccine itself, but, you know, how we go about specifically for this state to take care of the logistics for the mRNA vaccine, because it requires a different lift than most of our normal vaccines. It's a lot more delicate. It requires higher refrigeration or lower temperatures to transport. So maybe in the future. And then thank you for posting that Nature article for everybody to have access to. Any other questions for our fabulous presenter, Claire? I guess you got off pretty easy, Claire. You had some really good questions, and really thank you for your time, your thoughtful presentation, and your expertise in this, because definitely this added a lot to our understanding. If there are any other comments or thoughts before we close up for today, any alibis or comments on specific modeling that we presented on our data points, I'll leave it open for about a minute left, and if not, then we'll close up shop for this Thursday. And please reach out to us via our email for feedback, or if you know me and you have my personal email, Hawaii email, please reach out. We're constantly trying to improve our format and our reach. And if you have ideas on future deep dive topics, please let us know. We're here to support you and the public and our state. So it's a two-way street when it comes to this. So if that's all, then have a great rest of your Thursday, and we will see you guys in a week. Take care, everybody.
Great job, Claire.
Thank you.